honor and a pleasure to have her visiting. And we first apologize because this is the third time we invite her, and we canceled 2 times, so sorry for that.
It's okay, it's okay.
So Anna is an expert in proteins, anything that has to do with proteins and related systems. She made the postdoc with Alan Gosick in the States, expert in proteins and other things. So also a postdoc with my group in Madrid back in the old times, back in the good times, and now, hace aquellos years, I was working in Sevilla, now in the Centro Andaluz de Biología del Desarrollo. Interesting place with a lot of interesting activities in many different areas, for example, in different areas. And she's going to talk about language models. What else?
What else, right? So thank you so much, Alfonso, for the invitation. I'm very happy to be here and see known faces. Most of you don't change with time, so you have to tell me how do you do it. Because it's not working for me. So today I'm going to talk about language models. What else? No? Everybody is doing this nowadays. But I just want to give the message that maybe they are useful if we decouple for things that we know, like evolutionary conservation, right? Why is that? You will see soon. This is the situation we are facing with the sequencing data that we have now, okay? So this arrow will be like us trying to make sense of all the data that are incoming from sequencing projects. And we know that this resource, it is called Logan. I don't know if you have used it. It contains kind of a summary of maybe 2 petabases that you cannot see here. It's like 2 petabases from 44 petabases that contains the assembly of 27 million DNA and RNA sequencing data, okay? So there is a huge amount of data that we have to use, right? This is basically the size of the data set. So Ryan has made it available to all the community. So we can actually access this kind of data in a nice way. Now, the thing is that we need to, it's not working, so I will go here. It's not working either, so.
With a click.
You did something, eh?
With a click.
With a click, okay. So this is what I was saying. We have a lot of sequencing data nowadays coming from different sequencing projects all over the place, ERCA, Earth Biogenome Project, and so on, because we want to understand the biodiversity of the organisms and the functions in nature, right? So we have a problem, and the problem comes from the standard annotation pipelines that we use to annotate proteins of unknown organisms. All of them come from these model organisms, us, mouse, flies, worms, and so on. And the problem is that the experimentally validated fraction of this information is very low. And probably this number is getting lower and lower and lower by the time, right? So the thing is that it's not only few availability, the problem is that it's a very circular problem, in the sense that we are kind of transferring information from few organisms in a very few controlling situations to other organisms that are more or less similar, and then we are trying to transfer everything to the whole biodiversity. Okay, this doesn't scale, okay? This is a paper that I strongly recommend you to read from Armaral group. They made a study, very interesting one, trying to understand why we are studying the same genes all over and over and over and over again. And they identified promising genes that remain ignored. Why? Because people are not interested, because the scientific community is not interested, the fundamental agencies are not interested. But this is limiting our knowledge about the real reach of functions, right? So we are in this kind of framework now, the Groundhog Day, that we are transferring everything that we know all the time. So which kind of novel discovery can we get out of this? Very few. So let's revisit the basics. What are the basics? And I will talk before 2020, everyone knows why, okay? And we have three main characters here: sequences, structures, and functions, right? So before 2020, we could more or less know how to guess functions from sequences. We could get, more or less, structures from sequences. We could not get so much functions from structures unless you were in the enzyme world, okay? And then back in 2020, you know, that what happened, almighty AlphaFold came, and then it evolved everything, you know, we're solving all the problems and so on. And we could actually get models of every single protein that we want. However, this very important discovery triggered a very important conversation by Francis Arnold. We should move to protein function, right? Or maybe functions, right? So this is kind of a discussion that was going on all the time, and we still have this problem. We don't know exactly what function is, right? Baker, who I just got this very more realistic image of himself, you know, he kind of addressed the function thing by protein design, okay? But he was not interested in the function. He was interested in optimizing a function for solving particular problems, okay? But we cannot understand what is the relationship between these three kind of components, right? So despite these breakthroughs, when you go to function, then you cannot say anything about the sequence. Well, you can say very few things about the sequence. If you are starting from the functional point of view, you can say something about the structures, and this relationship is very unsatisfactory, right? Because we don't understand completely how they relate to each other, right? So we came with this simplification in the field that one sequence, if one structure, gives one function, right? This is the paradigm. And the thing is that this paradigm is at the heart of every single computational method to predict function, to annotate sequences, and to run functional genomics. I mean, what are the genes that are overexpressed in my RNA-seq experiment, for instance? So we get a problem here, because function is the elephant in the room, and no one wants to look at this in this view, right? So we know, in addition, that you may have two proteins that are interacting in a way, and it's giving you this binding mode, it's giving you a particular function. But it could also interact in a different manner, okay? And this one is giving you a completely different function. We have seen it in collaborations with Eva Esteban, a neighbor of yours. She's a crystallographer. Talk to her, she's great. And then we have same sequences, same structures, different binding modes, and different functions in different contexts, okay? We have also more challenges, like these typical moonlighting things. This is maybe the most striking example. So you will have the epigenetic histones, which are regulators of chromatin, and then they tetramerize, and finally they become an enzyme. I mean, we are talking about histones that are coupled reductases. This is crazy, right? Well, cases like this are coming up and up and up. And then we have this challenge. You may have exactly the same sequence that switch the fold, switch folders, in order to do different proteins, okay? Example, classical example, the CD terminal domain of this RFAH, you know, that is involving either transcription or translation. Same protein, same sequence, different folds, two completely different functions that you really need for a cell to live. So you cannot knock out one of them, okay? So in addition, when you move into other scientific communities, like genomics, Evo, developmental biology, they all believe that we have all the protein function figured out. I mean, they are strongly believe that we know everything about proteins, because, I mean, come on, it's only 1.5% of, you know, the contribution, who cares? So the real important thing is the genome. So they don't care about proteins, but they are using all the protein information in order to do their annotations and their functional analysis. So, I mean, they think they know, but they don't know. And they don't know. And I am so tired of telling them, no, you don't know. You have to work more on this. But it's very difficult, because this is still the elephant in the room. And it's the elephant in the room because we came with a very simple solution. This solution came from Google back in the 90s, okay? So they just believe that evolution is kind of a proxy. Evolutionary conservation is a proxy of functional conservation, okay? And I have shown you that there are cases where you don't have this kind of framework, okay? So everything was super focused on the identification of orthologs, which is itself a very difficult problem, okay? But if there is an ortholog, it's gonna do the same. We know this is where it's called the ortholog conjecture. But what about this? You have these two species, okay, with the common ancestor, and then you will have the ortholog gene in black, and then you will have a duplicated gene that is supposed to diversify the function. But we have many examples that challenge this, okay? These examples are challenged by functional analogy, and in most of the DNA repair pathways in yeast are made by proteins that are not orthologs to humans, okay? So this is functional analogs. And then you will have paralogs that retain functions, and they jump over, okay? And they take over the original function of the ortholog. And this has been shown many times, okay? Especially in developmental genes. Okay, so we have these problems. And the problem is a problem of definition, because in the end, what we need to do is to test the function in the lab to decide if this function is doing this, if this enzyme is keeping the substrate and doing this reaction, and so on, right? So what did we do? So we came okay, let's make some kind of order here. Let's use GoTerms, let's just create a gene ontologies in order to put some order in our definitions and functions. And we came with this experiment, which is extremely biased because it's trying to organize the very limited information we have about the functional space. Okay, so the problem is that we have a very limited functional space that we are trying to organize in a way that we can computationally access to it using graphs, okay? So this is a very powerful way to represent relationships, and then we can do things with them, okay? But we shouldn't forget that the Go terms are assigned to proteins, not to genes, and people are working with genes, okay? So this is a different level of complexity. Imagine that you have your red target sequence, and then you find similarities. We are using similarities as a proxy to assess evolutionary relationship. And then we decide that if two sequences are too similar, then they are evolutionarily related, which could be true or not, okay? And then you have information only of one of them, the black one. So what we used to do is to transfer massively all these informations from the black member to the blue members. The blue members may be doing something different, right? But as we don't know, because we haven't tested in the lab, we are just transferring these functions. And then we know that within protein families, we've been working with protein families Alfonso has been working with them for many years now, that you may have different functions. And sometimes these functions can be identified at the residue level. Okay? This will be the case of the GTPases and the small GTPases, but we know that each subfamily do very different things in very different cellular contexts, okay? So you just cannot transfer massively the annotation of one to the others, because we are kind of making it wrong, amplifying noise. If we do it automatically, which is what happens in the databases nowadays, then you have your query target, then your query against your target, you're very happy because you get this very, oh, nice, so 80% sequential similarity. Oh, that's cool. And then the only information from the target that you have is a protein kinase domain at the C-terminal region that is going to be transferred there because it passed your thresholds, okay? But you don't check that there is not a C-terminal domain protein kinase there. So this noise is getting amplified and amplified and amplified with all the databases and all the data we have nowadays. So this is a big problem. And the second problem is the no similarity, the nothing to transfer, lineage-specific genes, things like this. And I'm going to focus on this one, okay? I'm going to focus on this one because the no similarity issue was an issue for Rosa. Rosa Fernández, Premio Nacional de Investigación de este año, joven, okay? She's working, she's a neighbor of yours, you should collaborate with her. She's working with weird organisms, very weird organisms, biodiversity, okay? So she got a lot of strange guys, and she wants to understand why is the biology of these strange guys. And the problem that she got is, enough. I am trying to annotate these proteins, and I only get half of the genomes annotated. I don't know anything about half of the genomes of these guys. What do I do? I don't know. I mean, what should we do? And we were discussing with Beers about this, whatever she called dark proteomes. Dark proteomes in the sense that there is a substantial fraction of information that we cannot make sense out of it, okay? And with Beers, which always give you an enlightenment, right? Then we were discussing that maybe we didn't understand because the methods that we have nowadays are very powerful, okay? Compared to the methods that we have back in the 90s. We have lots of data now that we could use in order to train models to understand things. So it has to be an assumptions problem, okay? So our take was, well, what the hell, just forget about assumptions, okay? We don't understand them anyway, so let's do something different. And what we did is to forget the sequence similarity thing. And then it was a time that we were working in GAFA. We know that GAFA, for some of you that don't know what is GAFA, GAFA is kind of a CASP, it's a contest in which you evaluate the performance of function prediction methods, okay? This is old one, but this is the last published. The new one is coming this year, and I will give you a little insight about this at the end of this talk. The GAFA challenge, we found that the methods, they don't work too well. They don't work too well. I mean, there is a marginal improvement, but they are not improving, and we have much better methods now. We have more data, so why is not working? Because of the assumptions, right? According to the you can see here in red, the machine learning methods or using machine learning base were among the top performers, but they were not performing really well, okay? So we were taking a look about which kind of methods are people using, no? And we know that from the 10s and the 70s, the transformers were coming, okay? So we're getting into place. And then one of the guys that actually started to use all these transformers was Rojas. Okay, Rojas has been always working in function prediction and things like this and using machine learning. So he moved completely and fully into this deep learning transformer thing, right? So we decided to test some methods. Which methods? Okay, let's try to get the 2019 typical convolutional networks. That was the method that was used, DeepGo. Okay, it is called DeepGo. It's a convolutional network based on protein-protein interactions, some kind of evolutionary information, and so on. And then we decided to use the ProTrans model. The ProTrans model is a very generic, all-purpose model for function prediction task. And then, disclaimer, I didn't do this model, okay? So these methods were done by other people. But, we thought that maybe we could benchmark them more properly because they did it in GAFA, and they did very well, according to them, okay? So those are the results for them. They are kind of getting the results among the top performers and so on. But we shouldn't forget that GAFA, okay, is an universal average, right? So they are averages. We don't know exactly if it works well in an average of all the universe and how it works in a particular organism, okay? Because if I want to annotate a particular weird organism, I want to get a method that is giving me reliable annotations. So we don't know how they will perform at the model organism. We want to use it in non-model organisms, and we want to do it large scale. And this is what we did. So what we did is a two-approach system. First, a benchmark of these methods in model organisms in order to select the best one for annotating non-model organisms, okay? So what we did is we focus on yeast, mouse, fly, and worm, the typical guys. We know everything, all of them. I thought this is what I thought, that we knew everything about them. And then we estimated different performance. We estimated the performance of different metrics, okay? The first thing is that the percentage of annotated genes of these guys I was puzzled by this. We still don't know anything about 17% of genes in flies. Flies, we've been studying flies forever. There are cell atlases of flies, transcriptomics experiments for flies all over the place. Worm, 30% of the genes of the worm, we have no idea what they are doing. I mean, those are the model organisms. People are using this to study diseases in humans and things like this. And we don't know much of the information there, okay? So what we did is we extract the Goa terms, okay, with this kind of evidence, okay? Evidence, that experimental evidence, trust laboratory statements, and so on. We didn't get all the annotations that were there, just focus in these ones. And then we compare methods. We selected a stringent high precision, low coverage, okay? Hammer, because I like it, I like Hammer, and you know, with Hammer since always, you know. And then we compare the predictions versus the annotations. And this is here more or less the summary. I will start showing everything, but focusing in worm, just for the sake of this talk, because the worm was the one that we have less information about this, okay? You can see here the UniProt one, okay? The UniProt will be the orange ones, okay? In green will be the language models. The Pro3T5 is Pro3Trans. SECVEC is an LSTM other method, okay? Hammer, and then Decoupling. And what you can see here is basically for all the organisms, for each category of the gene ontology, these methods will annotate more proteins by average, okay? They are annotating more proteins, fine. They produce way more predictions per protein, okay? I was kind of surprised by this Decoupling thing, okay? They will produce so much annotations compared to the other ones. And actually, they will not do very well. You know, they will not do it consistently among categories, okay? So Decoupling was starting to feel a little bit shaky. Now, we have more predictions. We have more proteins annotated. What is the precision? I mean, can we trust them? So we devised this kind of a schema in which we will look at the Go graph, okay? So if the method will guess the term, that will be a hit. If it will guess the parent or the son, will be close, okay? If it will be the grandpa or the grandson or some descendant or ascendant, will be related. And then you will have a bunch of unrelated functions, okay? Things that will go to other branches of the graph. And that will be, for instance, the unrelated ones, okay? Things that you will not expect to be there. Could be false positives, could be something that we don't know, right? So this kind of the schema kind of mimics the human behavior because Go curators are humans, right? And depending on your level of expertise, you are going to annotate a protein very, very in detail or very general, okay? So we just wanted to know how well these methods will do that. So if you color the annotations for each method according to this schema, then what you can see here is that the language models, Pro3Trans and SECVEC, were doing much better than the other ones, okay? Hammer, of course, will do great, but it will give you very, very little information, right? And then this is the same view of the same information. So you can say that almost 75% of the annotations were close, which is very good, okay? Now, if you remove the Hammer predictions from this picture, you will still have a good performance, okay? So they will tell you nice annotation, accurate annotation, accurate information, despite removing what we know about them, okay? Now, you have to look at these methods like heavy weaponry, okay? You will have, Hammer, 3 shots, it hits the targets, very precise. Okay, you can trust them, but you cannot discover anything new, right? You can look at the Go, like, it's shooting all over the place, you know? So by chance, it may hit the target, by chance. And then you can look at the Pro3Trans, like, okay, it shoots a lot, but it reaches the target most of the times, okay? So this is basically the space that we want to work on if you want to find novel things, right? Things that you know they are gonna work most of the time well, and it's gonna give you new things that maybe like false positives, but maybe not, okay? And I will show you later that they are not. Now, which kind of metrics? The informativeness. You can look at the information content, which is giving you an information where in the graph this annotation is being predicted, okay? So the larger the values, the more specific the terms, the shorter the values, the more general the term, okay? So this will give you that deep go is giving you very general annotations. Biological process, okay, very informative, right? No. Hammer will give you more information, and then the language models will give you more detailed information, okay? So in general, the broad trans model and the other model, the LSTM, is giving you more specific terms from the graphs. Now, semantic similarity is another way to measure how similar are the terms that are between two groups of proteins, right? So we did it for the three categories. And then when you look at the language models, the green ones, they are doing much better, okay? Well, it's not novel. We know that molecular function is easy to predict than any other category, right? So we did like this, you have these numbers of 1, but however, when you move to biological process and to cellular component, it's giving you less semantic similarity. So they are kind of hitting different places, right? Why is that? Well, probably it's a problem of the network itself. The biological process is a completely different network as compared to the molecular function. You may have a molecular function reaching many different biological processes. So the network, the graph is more sparse and it's more difficult to assess, right? And with the cellular content, we had problems here, okay? Because it's something that should be very easy to predict, right? It's in the nucleus, it's in the cytoplasm, but it is not. It is telling you that somehow our definition of cellular component, maybe we should revise it, right? Now, what are the scores distributions of these methods? Because these methods produce scores, okay? So you, as a user, you want to know if I can trust the score that this method is giving me. So first of all, they are providing scores that are not comparable to each other. I mean, you can see the graphs, they are not comparable at all. So what we did is just to distribute them in quantiles, assuming that the last quantile will contain the most high-gain scoring annotations. Again, they will be the best, more confident predictions, you know? And then when you compare the scores at different levels, at the hit, and I guess the term, close, they are in the same branch, related and unrelated, you could see that for yeast, for worm, you know, all the distributions were kind of in the medium scoring terms, okay? So you will have a lot of correct genes at low scores, right? So this is telling you that the scores is meaningless, at least in our system, in our performance. The same happens with the fly. Now, let's move to the mouse. What happened with the mouse? With the mouse, when you have a displacement of the curve at the hit level. What is telling you this? This is telling you that you have to be aware of the sister species effect. Meaning, I did the benchmarking mouse, I remove all the mouse sequences, I remove all the embeddings from the mouse, but still I had human sequences there. This is why I have this kind of displacement. Second message, if you are annotating a genome, remove everything that is similar to this genome from the dataset, okay? Now, what is the scores effect on measurements? Well, you can see as expected that for the semantic similarity, you will increase the semantic similarity if you get better scores, you know? For the information content, it was completely irrelevant. They don't care, okay? And for the number of proteins, which could be critical depending on which kind of downstream approach are you using, because you may need to have many annotations for many proteins, then it's kind of a trade-off, okay? So we just decided that if you keep half of the annotations, then you will be fine, because you will get more or less the same semantic similarity, increasing the number of proteins that is giving you the similarity. Now, is that useful for functional genomics? No, that's experiments. What we did from all these model organisms, we extracted all the transcriptomics experiments that were good, and then we replace the annotations from the differentially expressed genes with the predictions, okay? And then what happens is that after calculating the semantic similarity, I am showing you here the results for the worm. You can see that in functional dynamics, okay, down-regulated genes using pro-trans models in an RNA-seq enrichment analysis will still give you very similar information than the original annotations. However, the other methods will give you completely not related terms at all. Okay? That means that you can successfully annotate a whole genome with a pro-trans model, and if you're running downstream RNA-seq, whatever enrichment analysis with these predictions, you will recover functional information. This is very interesting for things that we have not annotated at all. Okay? So that was kind of the summary of our benchmark, in which we found that the pro-trans model worked very well, at least for the purposes of functional genomics and other things related. Now, back to the 17% of genes that we don't know about flies. I said, well, can we annotate something from these guys? And then actually, when you look at these guys that you cannot annotate, they are intrinsically disordered proteins. These are very difficult to annotate using homology because they are not homology to anything, so you cannot transfer anything. But the language models, for instance, were able to annotate some of those. And one gene was this one, GC, whatever thing. And the pro-trans model was giving you a meiotic cell cycle-related function. But the good thing of flies is that you have so many transcriptomics experiments that you can see where this gene is expressed. So we went to the transcriptomics data, and we found that actually this gene was expressed in the somatic cell of testis male germinal. So somehow these models are identifying the biology. I don't know how, but they are finding it, and I don't know exactly why, but I found it fascinating. So, okay, we can annotate intrinsically disordered proteins with this thing, okay? Now, back to the dark proteomes. We were happy with pro-trans. We decided to annotate 24 millions of genes from these 1,000 gene repertoires from weird organisms involved in terrestrialization, when the guys went from the ocean to the land, and so on. And then we annotated all of them, okay? And that was done by Rosa and Gemma in their labs. So we run the same things. Coverage, we have to check for coverage in the sense that they had to use ECNOG mapper. ECNOG mapper is kind of the reference standard. It's based on phylogeny, evolutionary conservation. And this is the kind of pipeline that you will use to annotate novel genomes, okay? But you can see that you can only annotate half of it, okay? However, with these models, you can annotate all of it. Then you apply all these thresholds, everything, things like this. But just to make the long story short, you could basically annotate, even if you were very stringent in the scoring system, you could annotate a very large fraction of these genomes in order to do soil biology. We checked for the information content that was very similar for the protein language models as compared to the DeepGo and so on. Basically, it recapitulated very well the data that we had in the benchmark, okay? So the semantic similarity about the noun fraction with ECNOG, that was a little bit lower, but you have to consider that we are kind of comparing very unbalanced data sets, right? So this was not very relevant. What we did is to do enrichment tests against the annotated fraction versus the unannotated fraction, okay? To check if we could identify biologically relevant and coherent data. So we use the tardigrades that they have. I don't know how many tardigrades they have. And then we annotated the 40% of the genes, that is, they are annotated, okay? And then when we did the enrichment test, we found a group of genes with annotations that were significant in detection of a stimulus response to VBA, water deprivation, osmotic stress, something that is very, very related with the biology of these guys, because these guys are ultra-resistant guys, right? And now the good thing is that we have the genes. We have the genes that are responsible for those. And these genes could be very interesting because they could, help to develop, therapies for resistance to radioactivity or things like this, right? So now they are selecting a bunch of genes from this group, and they are running the experiments, okay? So the ProTrans annotated this fraction provides coherent annotations to biology, and we could even get the genes responsible for this function, something that it was not possible, and it's not possible doing it with annotation standard pipelines. We developed a tool. I mean, the tool is basically available, but we just made it for larger scale use, okay? And now we have made an update, including other models, CS2, PROST-T5. I tell you that PROST-T5 works worse in order to annotate function, because you are putting the constraint of a structure, okay? And well, this is available for you to use if you want. And what about the switch folders? The switch folders, I told you before that we have these cases where you have the same sequence, it changes completely the fold, right? So this is an example of a switch folder. This is the XCL1 protein. It is a GT-based activation protein in its monomeric state, okay? But when you run it to AlphaFold, okay, you will get the model. And the good thing of this is that AlphaFold was getting confused. I mean, AlphaFold should be capable to retrieve a very good model of this, but it didn't. So it is getting confused somehow. Why it is getting confused? Because this protein has a completely different fold. When it dimerizes in response to virus, it changes completely the structure. And AlphaFold is incapable to get this structure. It's incapable because he has to take a decision. Where do I go from now? Okay, I will go there, but I will give you a lower score. So it's kind of getting confused in places where you can find different changes, okay? So we run sequence-based and deep learning methods in order to get annotations for this protein, okay? And what you got is a protein binding receptor agonist, okay? You get all the annotations for the first conformation. And that will be the biological process. Taxis, organic, well, very, very general, right? Now, when you use the language models and you get the annotations for these language models, you get the CCR chemokine receptor binding, which is exactly what it does, okay? You get protein homodimerization, which goes to the function, molecular function is binding to itself. You know, this annotation was not retrieved previously. And then when you go to another, you get response to virus, which is basically where this structure was obtained in a response to virus, okay? Regulation, GT-based activity, and G-coupled receptors. And if you go to the graphs, there are terms very far away in the graph. So somehow, language models are capable to retrieve alternative functions, even if you don't have the structures. And they can tell you, okay, this protein is maybe doing this thing, something else, and so on, right? Now, the GAFA5 recap that ran the last year, so it was presented in ISMB the last July, okay? That was Kaggle enabled. So Kaggle gives you money. So when you give money, people tend to participate a lot, right? So we had a lot of teams, and we had a lot of methods. And just to let you know that out of the 10, the 6 top, 5 were using language models. So language models are here to stay. So either we understand what are they doing and why are they doing what they are doing, you know, because this is basically the future. I mean, we shouldn't rely in evolutionary conservation anymore if you are working in annotation corpuses, okay? And I mean, I hope I have convinced you that this is what we are doing now. For instance, in ERGA, in the ERGA consortium, we are going to start annotating with language models. We are not going to do it anymore. Okay, so this is something that is a change. Of course, there are problems. Of course, there are false positives, but of course, they are giving you a lot of information that you will not have otherwise, right? And just, I want to thank the people that have been involved in this development of this thing. I mean, the lab of Rosa, Gemma, my lab. We get some funding also for enhancing this tool and to make it like the de facto annotation tool for annotating biodiversity. And thank you for all your attention. So, do you have any questions?
Thank you, Anna. Any questions?
Yes.
If
You will have to shoot.
if we still need to search for proteins and we want to use sequence similarity, would be better to use just global alignment?
Well, it depends on your purpose.
Maybe you want to repeat the question because they are
Yeah.
following this on the.
So you say that if I use global alignment, if I want to annotate the function, if I want to use evolutionary biology. That was your question, if I understood it correctly?
It is more simple, but if we want to annotate based on sequence,
Okay.
it's easier to and more accurate to use the global alignment rather than local alignments for the tails that you mentioned.
It depends on your question of what are you annotating. I mean, are you annotating a particular region of your protein? Then you will go for a local alignment. You want to have an overall similarity, then you go to global alignment. But this is a different question. I mean, because you are assuming that an evolutionary relationship. So if you want to find how in evolutionary families you have a diversification of functions, then you can use your global alignment, get all the annotations from each of the individual sequences, and get how well they distribute along the Go graph. This is what we are doing also now, to check what is kind of the pervasiveness of multifunction in different protein families. But I will also recommend always using global alignments unless you are very interested in a particular region. Salva, yes.
Thank you for the talk. I really enjoyed.
Thank you.
I have two questions. The first one is about what role of plants. I was missing Arabidopsis
Yes.
as a.
Aureliano Bombarelli is doing this, okay?
So then
Okay.
the second one, would it be possible to use language models to evaluate existing annotation, like, well, human, we misannotate this number of things.
Yes.
Or even propose to remove them or something like that?
Yes, yes. This is something that the people in ERGA is doing. Okay, so they are kind of re-annotating the genomes, and from the genomes are getting the coding fractions, and they are revisiting the actual status of annotations in order to kind of refine them or so on. And according to the plans, Aureliano Bombarelli, who's is in Valencia, and he's working in a structural annotation of genes and also in proteins. So he's using this tool as well.
I'm curious about the plants.
Plants are problematic because, you know, plants have long genes. And we all know that the ProTrans model have very few, you know, a limited size vector. I mean, you can compensate for this, but there are problems because of this, you know, but you can use other models in order to increase the size or change a bit forward. You know, you can do techniques in order to solve this problem. But this is what he's doing now.
Thanks for talking and stuff.
Thank you.
So I'm curious about the generalizability of the models. So in the, let's say, training testing process, if you train on some species, test on the others, do you get similar performance, or it's something like stuff like that?
So to answer your question about generality, yeah, the question is about the generalization of these models at the subspecies level, right? Or at the species level, or?
I'm thinking, like, if you're training on a subset of species and testing on the same ones, like if you are now in a non-model organism, how accurate are your results? Because that's something that, of course, cannot assess without the patients, but.
Well, we just did the benchmark in model organisms, okay, using the information that we have in hand. And then by testing it in a completely different organism, we don't know how well it will generalize in the noun functions because we were not interested in the noun functions. We were trying to discover novel functions, but my bet is that it will generalize well I mean, not all of them. For instance, ESMC, whatever the last flavor of, it's very unstable. So this doesn't work. So you have to make kind of, I don't know, merging different embeddings, different models. I mean, this needs more testing. But my bet is that ProTrans is a general purpose protein prediction task that works very well for most of the tasks. So, and it's very simple. The problem is that we don't understand well how it works. So you can create a language model. This is something that we are doing in collaboration with another woman who is working in explainability, to tell us why this ProTrans is giving us this information. At least you can make sense out of it, you know? So you have language models talking to each other, you know, asking them things. Mm-hmm.
So my question was about, you said discovering null functions, because actually if we knew only few percent of link between the genes or between the sequence and functions, does this mean that we do not know most of the functions?
Exactly.
So we do not know biology.
Well, exactly. that's it. I mean, my question here is more he's talking about your question about the limitation of the non-functions, okay? So I totally agree with you. We know what we know, and it's very limited. The space of functions that we are using is very small because we have not tested all of them. What I meant by discovery is if I can find a function that is coherent with the biology of this guy in a group of genes, okay? That you could not find using homology because these genes are not similar to anything. All right, so yeah, we are still transferring the same kind of thing, the same kind of knowledge. It's not going to appear a new function out of the blue in the graph. You know, there are people working in this. They are kind of inventing new Go terms, right? People working in text mining and so on. But this is not what we are doing. But this is something that we should augment the data with potential hypothetical functions that we don't know of and see what happens.
Camila.
Thank you for this one. Great. I was wondering if you made any attempts to understand how the language models are doing their classification by looking at attention weights or something.
Yeah, this is what we are doing now. So
they learn also the phylogenetic information. I mean, somehow through their learning.
it depends. ESM does, but ProTrans does not. Well, does not, or we don't know to what extent.
You cannot know that it's not doing.
You cannot know. You cannot know, but I mean, you can try to know what it is doing. So if you are putting an attention layer, then you can direct the model to somehow.
I mean, this is back to your example with the mouse sequences that are predicted because they are
Humans.
But this would be true for anything because all the sequences are related by evolution. So it's impossible to remove all the evolutionary information by definition.
Well, it's impossible, but you are not using sequence similarity.
The system internally is taking the notations to the most similar one.
But we don't know how.
We don't know how.
We don't know how.
But it's impossible to have a data set in which you say there is no information transfer, like in the mouse model from the guys.
Yes, I get your point. I mean, we cannot say that this is not evolutionary related because everything came from an ancestor, right? But under the functional point of view, it doesn't matter. You can do many different things if you even are related to each other, okay?
Yeah, but then you cannot discount the mouse results.
Yes, you have to discount the mouse results because they are too similar to humans. So the embeddings, probably the embeddings from humans and for mouse are very similar.
Put it in another way, Anna. You will need to have a way of weighting the annotations by the score of the given level of similarity with another annotation.
Yeah, that could be done. I don't know if somebody has done it, but that would be a very interesting.
In this case, you're saying the mouse is very similar to the human, so I discount this because it's less.
What we do is we just you just run, you get your proteome, and then you discard everything that is over 90% of sequence similarity, and then you go through, which is kind of an mega ultra simplification, but you could use this information in a more refined way in order to learn things. That's for sure. Definitely.
That was just a comment because a few months ago here we had a lecture of machine learning, the same BSc of language models, and actually a model developed for one language is applied for another language.
Huh.
Because you have intrinsically the same way of thinking, and
Mhm.
that's a concept. We do not know how we do this, but
I mean, this is, it's a good point, and it's
Yeah.
actually applicable to that. When they train a language model and they add a new language that has not been in
Mhm.
the training set, the other languages improve too.
Yes.
So this is very mysterious and it's a little bit dangerous.
It's a little bit dangerous, and also, I mean, but it's basically the same concept of data augmentation. When you need to augment your data because you don't have data enough to run statistics or whatever, you just invent the data. But I think it's more a question of exploring all the theoretical space of things, of sequences that we are not exploring, they are not in nature, you know? I don't know. I mean, the thing that really worries me is that we don't understand how these things are happening. You know, and even if you put language models talking to each other, and the language model is explaining the output of this other language model, you don't know why this guy is kind of detecting the other thing.
but technically there is a mathematical ways to detect these correlations, I mean, and layers, no? Like
Yeah, but
vector
sometimes the
population.
models are so complex. There are so many hidden layers. There are so many operations that you cannot. In fact, you need another language models to interact with the other one. And this is where what explainability is doing now, with all the sharp models and all these kind of things.
And this is another discussion.
Yes, this is another discussion.
This is another discussion.
Hmm.
I don't follow.
Thanks for the topic. I was wondering, I mean, I use language models in a very naive way,
Like me.
trying to assist me to write reviews on topics on which I have worked for more than 10 years, and I have to revise very carefully everything because they make mistakes.
Of course.
How, I mean, these languages, if I'm talking from a biologist point of view in the bench, they want to use it to predict a function.
Mhm.
And if the function agrees with what the previous methods have done, it's easy. Okay, there's an agreement. Yes,
Mhm.
good. What about the new functions that are not discovered or described by the previous method? How can a biologist trust these models if, you know,
You have
it's
to.
very function is something, maybe the most complex part of biophysics.
You-
What else? I mean, how can we trust these models if we are going to write a paper and try to assay a function in the bench?
You cannot. You have to
Okay.
test them.
But
I mean, the problem, another way of
it's
saying
just
that,
a
the problem is that
contextual.
the problem is there is no scoring function.
Exactly.
All the problem of all this area,
Yeah.
whatever this is for text or for images or for proteins, the problem is what you're saying. You don't have a reliability value. You don't know how good are the predictions. This is why the protein structure prediction has been very successful because they came with a score, because you can calculate a score. You don't have to crystallize all the proteins to know that if something say 90% is 90%.
Mhm.
And we don't have any scoring, and explainability is not going to solve that because that will give you some reasons why there's something second, but will not give you a score. So that's the real problem. And this is why the language technology has been very useful. Has a lot of has a very strong technical limitation that cannot be solved with the language model. You need a different technique.
Yeah, but I mean, but it gives you a lot of information in places that you didn't have information before. I mean,
Generate hypothesis, but then you're back to what
Yeah, but
Paul is
the
saying. Then you have to test. You have to
propagate
test.
all these inferences to the node databases that people just query without really questioning them. Because if you have a, for example, let's suppose cross-trans is applied to the EBI databases.
yeah,
And then people trust blindly these databases, and then they transfer completely without questioning.
but it
the-
happens before language models.
Yes, that's right.
I mean,
So
you have data sets that are garbage that had been, propagated since decades in yeast and to hybrids and things like this that were full of false positives. And that was before protein model. So it's a more a philosophical question that how can we make a control and the quality of the data that we are using and we are producing? Yeah, I agree completely with you.
Do you think are there any consortiums, for example, for Drosophila, to try these new predictions to see if perhaps this is true or something? Do you think it's something that should be worth?
That should be
funding?
worth. I talked to all these muscologos, these flight people at my institute. There are a lot of them, And they are totally fascinated by this, you know, but there's no money to do that. There's no money to test this. I mean, who cares about this 20% of the genes if I am interested in the neuron, whatever thing? So I think it should be a consortium, a global effort that is trying to, I don't know, give some biochemical or biological meaning to these predictions, because then we could trust them, you know, but I don't see this coming.
This discussion about the annotations, it is an historical one.
Yeah.
There's always this by hand or by machine annotations, and nobody cares about, and everybody's using everything. So not all the problems are of the annotators. The users have to look at stuff.
Exactly. Exactly.
What is the role of kappa in this scenario? Because they are a consortium of functional annotation, and they're ranking models, and CASP grew like this.
Pues kappa is similar. I mean, it doesn't have the reach or the, you know, the impact of protein structure, because people are less interested in function. And this is because people thought that we have all the function figured out. There's no interest. This is a very old problem. Nobody cares. I mean, I do care, but I'm old and nobody cares, you know? People just want to do other things. But this is a problem, because you are using this information for many things in biology, and we are kind of running all over and over.
So what is the kappa approach then?
The kappa approach is try to monitor how well the methods are, you know, I mean, how well or how bad.
They are blind predictions in a sense.
They are blind predictions. So kappa with us, I mean, they do test. So they provide the competitors with a sequence that they have to predict, but there is a biochemical lab testing these process. Okay. Yeah. So in the end, and then there is some kind of metrics in which tells you accuracy and things. And if you, but they use gold terms in order to do this kind of.
Yeah, but it's as good as it gets.
Exactly. It's as good as it gets. You just say that. So
I give you the other possibility, the other view of the world. This is the more engineering view of the world, the more synthetic biology view of the world. This is too complicated, because proteins can do all these crazy things, do one thing in one side and then do something else with other proteins, or change function, or change structural function, or moonlighting, or something like that. Forget about that. Let's do this from the beginning. Let's do proteins with a single function and a well-defined sequence and structure, and then use this not only to have useful things, but use this as a building principle to understand how living systems are working.
Yeah, but for this you should sample all the theoretical space of sequences.
Well, you do what you can. You start somewhere.
And this is
you
impossible.
Start building a small microbe, and then you try to do something more complicated.
Yeah, but if you're interested in multifunction.
No, but they this. No, you can be interested in anything, and they will not change. People can do anything. I said that there is another way of thinking on that this problem is impossible to solve. And let's forget about that.
Forget about that.
Protein with a single function, and that we can use them to build systems, more complex systems than single protein. Because essentially, evolution is falling all the time in local minimum. These organisms will use this protein for something different than the other one, because just happens.
Yeah, exactly.
And it's impossible to disentangle that. So forget about.
But do you think you can explain all the complexity using a single function framework?
Sure.
I don't think so.
Sure. I don't see any reason why you cannot take all this very complex biological system and divide this in a smaller function that will be interacting one with another. This has happened by pure chance, because evolution was that way, not because it's intrinsically needed for anything.
Well, but evolution, it took a path because it was the optimal path to take at this particular moment, given the conditions.
It's the local path. It's the local path for this organism at this point in time. It's not the global optimal for anything. And this we know very well, it's not the global optimal for anything. In a different situation, something else will happen.
Yeah, but in the same situation, you could have many possibilities. Or not.
No, but no, that's another discussion. But put it like that, there's I think for the sake of the discussion, there is another way of thinking about that. That is going to be, that is intrinsically impossible to disentangle all the functions for all the organisms in life on Earth, because each one of them corresponds to a local minimum in which the same protein that you have been seeing this everywhere may be doing something different.
Yeah. Totally, I totally agree with you. But I'm more interested in proteins that being in a very precise context, like transcription, could start to translation. To me, this is absolutely fascinating. How can this happen? So this is a different problem. So to me, I just want to understand why, but I don't think I will see
Scientific
them.
curiosity is very well.
Exactly.
Any final question? Well, we thank Sarah for the nice talk.
Thank you. Thank you.